to a- ask about visitation, but also number of flowers, we chose a set grid that we went every 2 weeks. And working with the Forest Service botanist, identifying the flowers that we observed there across the season. And so we would walk that grid, count the number of floral resources that we saw every 2 weeks, which species was blooming, but we would also collect bees visiting those flowers. And these bees included endemic native bees, the ones that actually live there. And so we also put out pan traps to collect the endemic native bees. This is the first time a survey had been done for the Bear River Mountains.
From the Oregon State University Extension Service, this is PolliNation, a podcast that tells the stories of researchers, land managers, and people like you who are making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Malathopoulos, Associate Professor of Pollinator Health in the Department of Horticulture. There is a lot of concern about the impacts of honeybee competition on native bee populations, both here in the U.S., but also internationally. The thing is, it's actually quite hard to do the experiments to demonstrate this competition. So I was super excited to talk with today's guest who led a multi-year project to assess the effects of competition. Dr. Diane Cox-Foster is the research leader of the mighty USDA Pollinating Insect Biology Management and Systematics Research Unit in Logan, Utah. Along with her superstar team of doctors Kelsey Graham and Jonathan Koch, they designed a set of experiments to test the extent of competition under a worst-case scenario where bees were stuffed in a cage together, but also in a field-realistic scenario with a commercial beekeeping operation. So without further ado, let's head into the complex topic of honeybee and native bee competition this week on PolliNation. Well, hi, Diana. Welcome to PolliNation.
Hi, Anthony. I appreciate you asking me.
Well, I know a lot of people have been asking to have you on the show, really excited about some of so many wonderful projects going on at the USDA Bee Lab in Logan, but one in particular, I think beekeepers have been seeing you present at the conferences and they're very interested in it. It's this research around honeybee and native bee competition. Now, I know this is a topic that is of international interest, and maybe just to set it up for our listeners, like what are the mechanisms by which, a honeybee might compete with a native bee?
So that's a great question. So one of the things that we need to think about are for all the bee species that are out there, whether they be honeybees, bumblebees, or solitary bees, the critical, essential things that these bees need for their reproduction and for their livelihood are pollen and nectar. And so that pollen and nectar comes from floral resources. Depending on how many flowers you have out there and the types of flowers, you may see that the bees may all be trying to get the same resource. And one of the things that we know has been happening in the United States and other countries as well is that the number of floral resources that we have has been declining, both due to land use changes, increased use of herbicides, and then weather also is impacting floral resources. So as temperatures get hotter or you have increased drought, or on the opposite side, increased flooding, you have fewer flowers. So that's part of the reason how you can have this competition coming in. We also have beekeepers looking for bee pasture, places to put their bees for floral resources. In times when they're not out in a pollination event or just looking to something to feed their bees. And so you may have added colonies being brought into an area.
Well, that makes sense. So competition can occur because there's just limited flowers and a beekeeper may come into an area with limited flowers. And so presumably or hypothetically, competition may occur because there's too many bees for too few flowers.
And so you may want to think of that as carrying capacity. And the way that I explain it is if you have a room full of people, let's say 100 people, and you have ordered out for lunch, but only 10 sandwiches show up, and you're going to have a lot of hungry people in there. You're exceeding the carrying capacity for that limited number of sandwiches. And so by bringing bees in or by getting rid of flowers, you're actually changing the carrying capacity. And then we also need to think about, are there other organisms out there, i.e., mammals like cows, deer, elk, sheep, that might be wanting to eat that floral resource as well. I usually, when I talk about it, show this platter with the flowers on it, thinking you have a certain amount that are there. And that amount that's there is also affected by the weather conditions. And so you have to think about what's going on in terms of getting into those flowers too.
I've heard also people talk about concerns, not just about the carrying capacity issue, but that there may be pathogens being shared amongst different bee taxa. Talk about that a little bit?
Yeah. So let's talk about pathogens first in bees. So bees are just like us. So you have a wide variety of pathogens, bacteria, protozoa, fungi, and then you do have viruses, and you may have some parasites as well. Luckily for all the other bees, Varroa destructor, the parasitic mite, is only found in honeybees, so we don't have to worry about it being shared. So that's a great news. But for these other pathogens, it's primarily the viruses that seem to be able to move or be found in a lot of the other bee species. And there's a host of different types of viruses we know out there, and there's many others that we're still learning or discovering too. So understanding what the host range is and who's infecting who is important. And the way that we have this sharing of pathogens is unfortunately through visiting the flowers. So when I was a faculty member at Penn State many years ago, we did research and found that bees unfortunately defecate when they're on flowers. And that fecal oral route is one way that you can get pathogens shared. And it's not just viruses, but a whole host of pathogens like to glob onto pollen. You have to think of pollen being a magnet holding onto those and potentially being a route by which pathogens can be shared. So
So
people,
yeah, so I suppose the one thing about it is as well is that it has to be that the pathogen has to be able to infect another taxa, I guess. It might be
right.
poop there and it eats it and may not have an effect, but on another might have a worse effect, I guess. We might not know that.
Yeah. And that's where a lot of the unknowns are. Just what are the host range for a lot of these pathogens? What are the actual impacts when that bee is exposed to the pathogen? Is it getting infected? And what impact does that infection have? For instance, for you and I, we do probably encounter a lot of viruses that we get infected with, and it has no impact whatsoever, and it goes away. So even with honeybees, I liken these viruses being like little stealth pilots, that probably without having Varroa around, didn't really cause an issue unless you had other stressors coming in.
Okay, that makes sense. Okay, well, this is great. I'm glad we set that up, just like how competition might occur and how there might be negative impacts. Now, you got a project rolling a couple years ago. It's a great name, HIP-B, which stands for Honeybee Interaction Project. Tell us a little bit about how that project came about, how it got started, and then we can maybe talk a little bit about what the experiment did.
Yeah. So let me tell you about my research unit here in Logan, Utah.
Oh,
I
great.
have a I have a great crew of scientists and technicians, graduate students, and workers that work with me here. And our unit is unique. We get to work with all the bee species. And so I do have folks that specialize on bumblebees and can rear them up from wild caught queens. I have people that work on solitary bees, learning about how to get them out into pollination. And so we have a unique skill set. And so Project APSM reached out several years ago and asked if we would tackle this research question, given that we have the expertise to work on honeybees, the bumblebees, and solitary bees. And I cleared it with my superiors um, that we could do this. So Project APSM and Costco funded the study for 3 years. And this was a very big study. So I have to also thank my team members here in my unit. So Kelsey Graham, John Koch, Lindsay McCabe, Byron Love, and Craig Kunziger, and then technicians. And then we, through the funding, it was the only way we could do it. We're able to hire seasonal workers to come in to help us with this. So that's how it came about. And we came up with an experimental design. We went out and we asked multiple stakeholders across the board, beekeepers, conservationists, university researchers, many people as we could ask to provide input on our experimental design, to make sure that we were doing the best study that we could.
Okay, that's great. So that got it together, and you had this support, but also this broad kind of thinking about what an experiment to test for competition would look like. Why don't you tell us what that design was when, after talking with everybody and setting all the
Yeah.
wheels in motion, what were the questions and what was the design?
So the first question was, what does competition really look like? Because people had been talking about it, but it never really were set parameters or experiments to actually show that you could bring too many bees together, overdo your carrying capacity to impact them. So what we set up were cages. We have very large cages that we put over floral resources. And we either brought in honeybees by themselves, bumblebees, or a solitary bee by itself, or
Wait a
put
sec.
all
You said
together.
cages. What? How big are we? Like, are we talking a cage the size of like a conference room, or how big? How big are these things?
Maybe not quite that big, but
Okay.
20 by 20 by 10.
Okay. All right.
So fairly good size. And we had irrigation to support the flowers. First year we did it with just one flower and found out that solitary bees, as we knew, really love Phacelia. But it turned out honeybees and bumblebees, the colonies just declined over time.
Okay.
So why that is, but I think that's important. We found out that honeybees and bumblebees needed a diversity of flora resources.
Okay.
Solitary bees made use of them too, which is great.
Okay.
We did actually demonstrate competition when we brought all three bees together. So this is a full-size honeybee colony, full-size bumblebee colony, and about 15 solitary bee females that were mated with 15 males
Okay.
together. And we have floral resources that bloomed across the season. We would go in and monitor reproduction of the solitary bees, figured out how to peek inside the bumblebee colonies without disturbing them to figure out numbers. And in the end, we took apart the colonies to look at how much they had grown, how much bumblebee organic material, live bees, queen cells, honeybee colonies, we weighed them. Also looked at number of frames of adult bees.
Okay. Oh, good. This is great. So you know if there was competition, because if the colonies did badly, then
Right,
they clearly would. There's clearly competition. But if they didn't, they all grew independently at the same rate, then it would be. Or if one grew faster than the rest, you would say, oh, this one's competing at the expense of the others.
right. So we
Okay.
could ask about colony growth. So honeybee colony, for instance, grows and then you get swarming for reproduction. Bumblebee colonies get to where they make queens. They're growing and making queens. Solitary bees, we can actually count the number of offspring.
Okay.
Yeah. So what we saw there was that all three when they're by themselves, did well. We saw growth of the colonies and we saw lots of reproduction by the solitary bees. When they put them together, all three of them suffered. We didn't see any winners.
Okay. So they're at the expense of everybody else. Okay.
Yeah. And we also asked, what did they do on the flowers? So if my team went in, they would actually follow individual bees around and record what they did, what flowers they visited, and then also what type of interactions happened when the bees were on the flowers together. So we could ask if there was a bully, somebody that was defending the flower or shoving everybody off.
Yeah.
We didn't observe that. They all peacefully interacted on the flowers together. And we did see for the solitary bee, Osmia bruneri, that it changed the gestation for one flower species versus another. It went up and went down on one. But that we didn't see a change in the honeybees or bumblebees on those flowers. So we don't understand that. But the critical thing is we didn't have any bullies there.
Hmm.
And I find it humorous. We saw more midair collisions. I like to say that we needed a flight control person in there, to help direct flight. Yeah.
Now, clearly, this is probably like the highest. This is as high a competition as sort of the extreme end. But you did experiments that were more realistic, that were more in place. Tell us about those.
Yeah. So we had a cooperator, Darren Cox of Cox Honey Incorporated. He's now retired, but when he was active, he had active Forest Service permits. And so east of Logan are the Bear River Mountains. Part of its wilderness area and other parts are Forest Service land. And he also had apiaries on private ranches. And so working with Darren, we were able to find 3 parent sites to do our research. And the first year we focused in on this private ranch where Darren had had an apiary for over 20 years.
Okay.
The apiary had 90 colonies or twice as many every
Okay.
year. Yeah. And he only put 48 colonies in for the time that we did our research. And 5 kilometers or a little bit further, we put out the bumblebee alone apiary. And at the honeybee site, we put bumblebees as well. So you have to think of it looking like a bullseye with the honeybees directly in the center with bumblebees. The very farthest ring out has bumblebees alone. And then at both of those sites and 5 sites in between, we put boxes with the solitary bees that we seeded with females. And so our design was thinking if the honeybees are going to impact the other bees, we would see this impact dilute the farther out that you went with the solitary bees.
Oh yeah, because the honeybees will forage most intensely close to the colony, but by the time you get to that outer ring of the bullseye, the impact should fall off.
Yeah, and that 5 kilometers is well outside of any range that has been set for honeybee foraging.
Oh, that's why it's set at that. Okay, great. And so these honeybee colonies, and the thing I like about this study as well is that it's a regular size apiary, so 48 colonies.
Yeah, this would be what commercial beekeepers would want to make use of if they're bringing bees into an area. That would be an okay size. You have to think of their expenses and labor to get colonies in, and something that you could easily manage as
Okay,
well.
so it's very realistic in that way. And I suppose so the colonies were moved in and the bumblebees would be placed when the bumblebee colonies would be started naturally. And I guess
Yeah.
the, the
So,
Osmia species would be put out about the time that so you
right.
followed it over time.
Yeah. So our bumblebees were a native species, Bombus huntii, or
Okay.
the hunt bumblebee, that we reared up from wild-caught queens.
Oh,
And we put them out. And there was a cage inside we put it inside a larger box to protect the colony and give them shading.
okay.
Insulation. And it had a control access in there. And so we had previous researcher devised a pollen trap that we put on the front here. So that was the main way the bee
Ah,
in and out.
okay.
No, we didn't engage that trap unless we were out there trying to collect pollen. We wanted pollen from the honeybees and pollen from the bumblebees to ask what plants were they foraging from.
Oh, interesting. Okay.
that didn't quite work as well. The hunty-eye bumblebees were pretty devious and didn't always lose their pollen when they came back. But we did get it from the colony itself. We also put a sheet of acetate underneath the bumblebee colony that
Mhm.
allowed us to collect their fecal material.
Interesting.
And part of the reason we did that, we know a lot of the diseases, especially the viruses, are shed in the fecal material. So we collected that every 2 weeks for each of the bumblebee colonies.
Oh, amazing.
And for honeybees, we took bees at the beginning and bees at the end of the season to look at their viruses and that they had. And solitary bees, we sampled them in the offspring, in the diapause adults, to see what diseases they had.
Okay, so we can see the growth of these colonies. We've got estimates of the disease that they have.
Right.
I suppose the other thing is you sometimes hear, "Oh, well," I was thinking about your food platter example in the boardroom, that it would be nice to be able to see how many, what bees were going to which flowers.
Right.
How did you count that?
Yeah. So to a- ask about visitation, but also number of flowers, we chose a set grid that we went every 2 weeks. And working with the Forest Service botanist, identifying the flowers that we observed there across the season. And so we would walk that grid, count the number of floral resources that we saw every 2 weeks, which species was blooming, but we would also collect bees visiting those flowers. And these bees included endemic native bees, the ones that actually live there. And so we also put out pan traps to collect the endemic native bees. This is the first time a survey had been done for the Bear River Mountains. And we got over 9,000 specimens.
Oh, wow.
So it's going to be a really good record of bees across the season because the solitary bees, they differ in when they appear, emerge as adults, and interact. So we're able to get a full spectrum now for this region up there. Yeah.
Wow, that's a lot of data collection and a lot of just a lot of different features of competition. I guess our listeners are really interested to hear what the results were.
Yeah. So I need to tell you first about a surprise.
Oh, good. I love surprises.
And the surprise wasn't a pleasant surprise at first. So we worked really hard to get all these bumblebee colonies out there. In the first season, we just did the one site, this permanent site with this permanent apiary, because we wanted to work out the kinks in how we were doing the research. And this was a really big surprise kink that we found. And so came back about 2 weeks after we put the bumblebee colonies out. And my team went up there and they worked in the evening after dark because the bumblebees actually are more defensive than honeybees. And they sting multiple times and they don't forget. So we had to go after dark with red lights. And they found that all but one of the bumblebee colonies had been invaded by a kleptoparasitic bumblebee. It's called Bombus insularis
Really?
or Cithrus insularis.
Uh-huh.
And so this was devastating because the way that this bumblebee makes her living, she doesn't have the structures on her body to collect her own pollen and nectar. So the only way she can reproduce, find that pollen nectar, is to invade another species colony, another bumblebee colony, come in and kill the queen, kill any workers that are trying to fight her, and then enslave that colony to rear up her brood. So this wiped out our sentinel bumblebees that first go around.
Oh, my
So
God.
we made lemonade out of lemons, figured out how to put another excluder on there that would allow the hunty-eye workers go in and out, but keep the larger citrus out. So excluder. And that worked. And that allowed us to move forward for the next few years using Bombus hunty-eye.
Just as a quick point, there is a great episode that we had earlier. I can't remember the person in Pennsylvania, the researcher that looked into these excluders. If listeners are interested, just go back into the archive. There's a great episode on this. But keep going, Diana.
Yeah. So we then did an additional study on the Bombus insularis. So it is an apex predator. So in top of the chain here with the bumblebees. And there's other data out there for other groups showing that if you have impact on prey items for the apex predator, where there's population decreases or effects on the populations of the prey, you see that the genetic diversity of that apex predator goes down. It's actually being impacted. So we did population genetics on the citrus, asking about genetics in the apiary site where this had been there for 20 years.
Yeah.
With lots of honeybee colonies there, at least 90 or twice that amount.
Yeah.
We're out here at the bumblebee only site where there had never been any honeybees. And what we found is that there were equal genetic diversity at both sites and equal numbers of citrus at both sites. So there was no impact at this long-term apiary, at least from the previous year, or a long-term impact on the bumblebee prey items that the citrus was making use of.
Oh, right. And I just, as you said, as an apex, as a kind of like, it really depends on bumblebees for its reproduction. It is almost a better indicator of maybe the health of the bee community, because otherwise it couldn't support such a diverse and abundant population of these cuckoos.
Right.
Oh, that
Yeah.
makes sense. Yeah. Okay.
Yeah. So this was a surprise, but we worked our way around it, but we learned something from this surprise as well. And just as a note, that citrus was what they were trying to get in the honeybee colonies as well. So we would find dead citrus down at the bottom where the honeybees successfully killed the citrus too. But every year we would see these citrus at all of our sites trying to get into our bumblebees. And because we made this little tiny excluder, we were able to keep doing our research.
Okay, so that was the first year.
Yeah, I could put a plug in here for why this finding is important too for agriculture pollination using bumblebees. Some of my team have gone out in blueberries where they've used citrus or cranberries, and it's not clear that the bumblebee colonies there have an excluder that would keep citrus out. And citrus is a species that's found across the United States. So if you have pollen or groups wanting to do pollination with bumblebees, asking if you can get one of these excluders or figuring out how to keep your citrus out of your colonies will allow your bumblebees that you've paid dearly for to go out and do their job.
I remember the episode now. The issue was not that they had cuckoos going in, but because the colonies were so much larger at a time when queens were emerging, queens would go into these colonies because I guess even non-cuckoo bumblebees will try to take over the nest or enter a nest site, and they would just get killed. So it was kind of like, without these excluders, these full-size colonies coming into pollination would then end up becoming these magnets for queens that just...
Right. Now, one of the other things for those excluders, if you're using Bombus impatiens, especially in the West Coast, it'd be important to have that excluder on there to keep the queens of Bombus impatiens from getting out and becoming naturalized in an area.
Okay. But coming back to it, so the lemon's the lemonade. We got, so you weren't able to get bumblebee data in that first year. Well, you were able to.
We brought more colonies back. We brought more colonies in. We had enough backup, fortunately, that first year, because we were just doing that one site, that we could get more colonies out there and still move. Yeah. So that was good.
So tell us what happened. So I guess the question is, did the bumblebee colonies close to the honeybee colonies grow slower than the ones at 5 kilometers?
So the answer is no. We did not observe across the 3 years. So colony survivorship and colony growth varied a great deal by colony. So we put them out with equal numbers of workers, and we can't explain why one colony would do better than others, but it was not related to the presence or absence of the honeybees. And we also did not see an impact on queen production by the honeybees. So that wasn't there. This research also revealed to us that we don't really understand what's the likelihood of bumblebee colonies to live out in the wildlands or in the native areas. So we have a lot more research in the future to try to understand that.
I do remember old research that my former postdoc supervisor, Ralph Carter, had done with bumblebee colonies in southern Alberta. And just, a lot of those colonies that start never finish. That there's all of that, you assume these queens, just like a honeybee colony, just go all the way to fruition. But where that mortality happens during over the whole population, I think that's a great question that I don't know many people know about.
Yeah. So, and we may have a little bit more clue on mortality factors for bumblebees from our disease studies. But if I can tell you about the solitary bees in competition
Yes,
first.
please. Yes.
Our honeybee colonies, we did see about 10% that either had a queen event or became drone layers. And that's, I think, normal for most colonies, both in commercial operations as well as other hobbyist operations. So we didn't have exceptionally disappearance or die off by our honeybee colonies. So bumblebees, I told you about how they did. The solitary bees, I think, is most important. So there we can actually, each female solitary bee claims a little cavity as her own. And for ours, we had these boxes that we drill holes in and we could put paper straws and we fold those out whenever they got filled. Because we knew, and it was demonstrated here, that you actually have competition for those cavities by a lot of different bees, and including wasps that want to make use of it for their nesting too, predatory wasps.
Yeah.
And so we constantly were changing those out. We could bring them back and we could x-ray them to figure out who was in there and also count the number of bees offspring that each female created. So what we found was that for Osmia bruneri, we had equal reproduction, number of nest cells, going from whether you were at the honeybee's site, the center, all the way out to the outermost region. Amongst all three, there was no impact of the honeybees there.
Interesting. So no impact on the reproduction of bumblebees and the Osmia bruneri, not only across sites, but also in proximity. So the ones that were far outside the honeybee range did just as well as the ones that were right beside the colonies.
Right. Yeah.
Interesting. Okay. Well, that's unexpected.
Yeah. So for 3 years that we did it, we had different weather conditions. So drought is an issue in the West and high temperatures. And so the first year we had severe drought. Second year was extreme drought. And the third year is moderate drought. We found that extreme drought year actually had a biggest impact. It impacted the honeybees, it impacted our numbers of flowers, and it impacted the solitary bees. So on honeybees, because the flowers just stopped blooming, they came out earlier. But the overall honey production of that year was 1/3 less than the other 2 years. So it significantly impacted the honeybee colonies. The solitary bees, we actually found a surprise there that it impacted the reproduction of the solitary bees. So they made many fewer nest cells. Surprise was what happened in those nest cells. So the solitary bees, bumblebees, and honeybees, the queen or the female mother can decide whether to lay a fertilized egg that'll develop into a worker or a future reproducing female, or lay a drone egg, an unfertilized egg. we found in that extreme drought year that the solitary bees made many more males than they did females. The sex ratio shifted from 1 to 1 female to
Hmm.
3 males to 1 female. So we have many fewer nests or offspring, but many, many fewer females being produced. So one could predict that if you had this continued drought occurring, high temperatures and drought, you might see the populations of that solitary bee out in the wild go way down. And I think that was one alarming thing that we saw.
So but bringing this all together, though, there is a way in which all the bees uniformly, in their own particular way, that honeybees less honey, solitary bees sex ratio and lower output, they're all being affected by the drought. So the even more pronounced than the lack of competition, I guess what's pronounced is that all the bees uh, suffer in their ability to do well when
Yeah.
conditions are suboptimal.
Yeah. And for bumblebees, they did reproduce. We did see queen production in that. We did see increased invaders coming into some of the colonies. It wasn't, there were some honeybees that showed up, but there were other bee species that showed up too, and wasps that came in those bumblebees. Didn't seem to impact the ability of the bumblebee colony to survive and do things, but out for the other consumers of flowers, the other mammals that want to eat flowers, we did see some impacts there too. So one of our sites did have sheep on a grazing permit. They brought in 7 truckloads of sheep to one of our bumblebee only apiaries, really ate them, and then drove them up through the mountains. The sheep ate every flower
Oh,
from a type of mint that horse mint that was growing there. It was a very preferred plant for honeybees and bumblebees both, and other bee species. That plant never reproduced. We also saw in that extreme drought time that the cows that were out there were basically tromping through all the flowers, just trying to find anything to eat.
hmm
So we have to think about them as well in carrying capacity. The other component that we had, we did this during COVID, and my crew says it's not just COVID, but there's a lot of people wanting to recreate up in these mountains here. Some of them do bring in their all-terrain vehicles and ride up and down on the Forest Service roads. That increased traffic, we could see dust clouds coming up and billowing off the dirt roads and the dust going over settling on flowers. It's been shown by a lot of different researchers that dust and other pollution getting on flowers actually impacts the quality of the pollen and the amount of nectar. So we have to think weather conditions having a bigger impact, maybe also through this dust production on the quality of the pollen and nectar too.
Well, look, maybe we can kind of come back to this question of what a land manager, or, if you're a rancher or something, could do. But just finishing off on the results, you mentioned as well that there was some disease dimensions that you were. Tell us a little bit about those results as well.
Yeah. So my own
Yeah, and just
specialty,
to remind people that I think most people have in mind is that the honeybee colonies would be giving off pathogens that would then make their way into these other bee taxa, causing problems. So tell us.
yeah. So for honeybees that varroa mite, when it came in, we see this increase in the amount of viruses, the number of viruses and the amounts of viruses that we see in honeybees. And that they can deposit these viruses in their fecal material on pollen of the flowers that they're visiting. And I think we already talked about that there are other pathogens maybe ending up on there. So that's one of the concerns that a lot of researchers in Europe and also in the United States have expressed, that maybe the spillover from honeybees because of this deposition on the pollen would be an issue. And you can go in and find viruses in bumblebees and solitary bees. So with our data, we actually asked about impacts. So we're able to follow the honeybees at the beginning of the season to the end of the season. We follow the bumblebees, looking at their frass. We're able to recover viruses out of that. And in the solitary bees, able to look in them. And one good result that we found for honeybees, so these colonies were coming from PolliNation event in California, the almond pollination,
Hmm.
with a layover down in the valley of Utah. That's my scene behind me there.
Mm.
So they did directly pop out of almonds into the forest. But when they first came in, we detected a fair number of viruses. We were looking at 6 of the RNA viruses and basically finding almost all of them.
Hmm.
And we did quantitation of the viruses, figuring out how much titer of the virus, how much did they each bee have. And what we saw is from beginning to the end, by looking at colonies and matched samples, is that the number of viruses went down over the season that we could detect, but also the quantity of the viruses and the prevalence within the colony went down. So what it does say there, and it confirms what a lot of the beekeepers suspected, is that when you get out away from ag with pesticides and other stress factors, you're eliminating part of that stress on your bees. And so you see that they became more healthy. So that was cool.
That is really interesting.
So I'll tell you about the RNA viruses. So in bumblebees, what we found there is that, yes, we could find the RNA viruses both in wild-caught queens, but when we brought the colonies out into the forest, we could also see added virus coming in, both in the honeybee apiary, but also out here in some of the the bombus only apiaries, suggesting there might be other sources of viruses spilling over into those. When we
Oh,
look,
I see. Okay.
yeah. So there is sharing of these viruses somehow happening. But when we looked at what those viruses did, the RNA viruses, we saw colonies that lived, that they had high numbers of these viruses and they kept them all through the season, but there was no impact on their number of queens that they produced. Colonies that died, we didn't find that many of these RNA viruses there. We can't blame the RNA viruses for the death of the colonies. Or for colonies where the queens were lost in the bumblebee colonies, we can't blame that. And in our solitary bees, research done by my graduate student, she's now Dr. Mary Kate Williams, in Osmia lignaria, we now know that these RNA viruses, when we give it to the larvae, they lose it at pupation, and we have no impact on survivorship, development, or weight. And we saw it at Osmia brunneri, none of these viruses in those diapausing adults either. Through Mary Kate's research, we also found evidence for a DNA virus sequences in wild-caught Osmia lignaria. And so we started looking at it. We included that in Hip B. This virus was originally described in 1976 by an ARS researcher and found in honeybee colonies that were dead and dying. And he called it Apis mellifera filamentous virus. And it's a DNA virus. It's actually huge, both in size and how much DNA it has. And we found it in the wild-caught osmia. And in honeybees, we also found all of our colonies bringing this virus in at very high levels. And it's much higher than the RNA viruses in terms of titer. Like, it went anywhere from 100 virions per B cell up to 1,300 virions per B cell, which is unheard of. And those colonies still lived. And the virus did decline, but it stayed in those colonies. We also found it in wild-caught bumblebee queens. And more importantly, in our colonies that died out in our experiments, 80 to 90% of them have this filamentous virus. We think that it is a mortality factor for them, but it was not associated with them being near honeybees. They had the virus. It was some other stress or something affecting it.
All right, let me sum this up. So you've said quite a bit here. So the first thing is when you looked at just the viruses in general, the honeybee colonies they came in, over time, their viruses go down in this location. And then
Right.
when you looked at varying distant, close to the hives, far from the hives, and you looked at the solitary bee and the bumblebees, the patterns of viruses did not seem to be, what we expect, that the viruses are higher closest to the honeybee colony, suggesting, as you said, some other source, even though there are viruses coming in, it's not, one can't explain it merely as the honeybees are disseminating the virus. There may be other places the virus is coming in,
Right.
and that some of these viruses may have differential impacts, that in some species, like the DNA virus you were describing, in honeybees, it may not have as much an impact as it does on bumblebees.
So I think that we need to look at more. So this DNA virus hadn't been really studied a great deal. So 2024, there's 9 papers total. People saying it's a stress factor that causes virus. And so it can kill a colony and lead to paralytic-like events or poor metabolism. So one of my concerns right now, bigger concerns in terms of bee health, is just understanding what that is, because every honeybee colony that we've looked at since then, we can find this virus. So I think we need to ask, is it involved in colony losses? So, but leaving that aside, for the bumblebees, that yes, it can be a mortality factor, and we just need to understand more what it's doing there. So our wild-caught bumblebees were not associated with, or wild-caught bumblebee queens were not associated with honeybees. So we are finding virus in them. In the Osmia lignaria, we're finding it in wild-caught bees that are in wilderness areas where there's no honeybees. So this host range of this virus is much bigger than what we anticipated. We also know it's found globally, and we can't blame honeybees on it. But it does point to this virus as being of concern in terms of what stress might do to it and its infection impact on the bees. Does that
Well,
help?
yeah, no, that's perfect. Thank you so much for laying this out. This is a really interesting study, and I know a lot of beekeepers and conservation people are looking at it closely. And I guess maybe to end the interview, just tell us a little bit about, it is one study, a very comprehensive study. What is its significance for maybe looking at this problem, more generally? What are some of the gaps or caveats? And I suppose this also raises the question, if carrying capacity is so important, how does someone like a rancher, for example, assess their carrying capacity, or a forester? How do they know that there's enough colonies that can come in? Tell us a little bit about that.
Yeah. So just to lay it out, there are other researchers now in the US who are also reporting that they're not seeing a negative impact of honeybees onto other bees. But back to this carrying capacity, because I think there is agreement now that that is the critical thing, both in Europe and the United States. How do you evaluate it? And that's difficult to do because, as I laid out, weather's a big component of this. Weather's determining what your flowers are going to be. And so I think that's where we need to do a lot more research on that. But we've also been doing some research here with the other research unit using drones to capture images. And using the drone images, we can now get an estimate of the abundance of floral resources over a fairly large area of land. So we do
Oh,
have a
wow.
paper published where we flew the drone in the mountains for one of the hip bee seasons. And for 7 flowers, we're able to quantitate the amount of resources there. And so with our collaborator, Alexander Hernandez, he's in Forage and Range Research Unit here, we're carrying that forward to try to get that estimation out even more. And Alex is also talking about how he's developing potentially an app that you could use on your phone to capture an image and quantitate things as well.
Well, this is just another testament to, there are very few labs in the world like the lab in Logan that has the capacity to do bumblebee, solitary bee, honeybee study and do it so comprehensively. And we're all excited to see these findings as they come out as they get published. Thank you so much for taking time today to tell us about this research.
Thank you for meeting with me today, Anthony, and giving me the chance to talk about our research.
Thank you so much for listening. Show notes with links from each episode are available at the website pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's a form at the website where you can pop in and say hello and give me feedback. If you want to support the show, remember to leave a rating on iTunes, Spotify, or whatever podcast mothership you use. And finally, if you have the means and you want to help support my lab's effort to document bee biodiversity in Oregon, visit OregonBeeAtlas.org and follow down to the donate button where you can make a tax-deductible donation to the Jerry and Judith Paul Native Pollinator Endowment. Every little bit helps. See you next episode.